So we are going to take a little bit of notes that we have left for this unit. It's only 2 topics and pretty simple. That'll probably take about half the class period, I imagine. It's got mostly examples embedded. If you see, it's only 8 slides, so it's not even a lot. Of the 8 slides, only 2 are notes and the rest are examples. Okay. Then when we're done with that, I made a Kahoot review for the content. Your Unit 6 exam, so your stuff on Mendelian genetics, non-Mendelian genetics, and then this content is going to be next Tuesday. We're going to do the same format as last time, so you'll have your free response during class Tuesday, and the multiple choice will be due that evening. Multiple choice is not quite as long. I think I made it somewhere between 20 and 30 questions, so not 52. So hopefully that's a little bit better for you. I didn't add a separate something into today that had just College Board practice questions because the examples that are actually embedded here in these notes are College Board examples. Okay. And per usual, as we go over this, if you have any questions, let me know. Does anyone right now know of any questions they have about something I just blabbed about? Okay. So today we're going to talk about the last 2 topics. There you go. That's a valid question. They're on Canvas. What you should be asking, Nicole, is, are they published so we can actually access them? And the answer to that would be no. So if you refresh, they are now. I'm so sorry. Thank you. All right. We're going to try again. So our notes that we are taking today are on recombination frequencies and pedigrees. Pretty cool. The pictures I actually have on this slide with the gray background in the corners, those are actual microscopic images of chromosomes crossing over during meiosis, which I thought was, I thought was pretty cool, yeah, to look at, right? So you can actually see them overlapping, and you can really see in these top corners, you know, the random chunks of them crossing over. All right, so our first topic here is on gene recombination. So what does gene recombination mean? Well, it's when combinations of gene variations get mixed up. This occurs during crossing over of meiosis 1, like we've covered, and it's not necessarily an accident, right? It's useful because it increases genetic variation and diversity. And as we'll get to eventually in our last unit of the course, you want a population of organisms to have as much diversity and variation as possible because it helps increase the chance of the population surviving with environmental changes. Gene recombination, or the genes crossing over in meiosis, like we've said before, is completely unpredictable and unique, but it's also part of what guarantees that every single sex cell produced from this event is going to be genetically unique. We did. So some of the standards that are in their ecology unit overlap a little bit with concepts in natural selection. So we have talked about that. And I think inherently a lot of those concepts too, you know, when we discuss them, they just very easily make sense and are almost commonsensical, if you will. So what is recombination frequency? Recombination frequency then is what it sounds like. So we know what gene recombination is. Frequency is how often something happens. So recombination frequency is how often a gene is swapped during crossing over. Now, this information is useful because it tells us the relative position of genes in our DNA. And you might be thinking, cool, why would that possibly be useful? Well, when we can actually sequence our entire DNA, or what we would call our genome, it allows us to know exactly which regions of all human DNA codes for which types of traits. That is useful because it allows us to more accurately be able to pinpoint which regions of our DNA code for different types of disorders, health issues, cancers, things like that, okay? And it's also what allows us to eventually have developed things like viral gene therapy, where we are then able to inject people's cells with viruses that carry the correct variation of chunks of DNA to kind of remedy these genetic health issues, disorders, diseases, etc. So when we're looking at gene recombination and recombination frequencies, the closer the gene variations, the more similar the recombination frequency is. Okay, if they are further apart, the less similar the recombination frequency is. We're going to do an example with this to make sure you understand it, and I think the example will help you make sense of what I'm talking about. Anyone else in here get headaches always leading up to and during precipitation outside? I always do. And so I have one right now, and I just feel like that's highly offensive. Like, how am I possibly supposed to feel healthy by controlling the weather outside? I can't. Disrespectful. Right. Obviously, fatal. Cancel precipitation. I just need to go outside and be like, can you not today? I just, thanks. Yeah. Some of you probably get it and you don't even realize it. Like, you haven't put the pattern together yet. If you're someone who gets headaches frequently, you might just think you always have headaches. But once you eliminate stuff that triggers your headaches, it's very clear, like, anytime there's a huge pressure change outside, which is almost always followed by precipitation, or a huge change in temperature, I get a headache. And so the pressure has changed because it's supposed to rain today. It likely is. It likely is from the weather. Sinus headaches, they're usually ones that you feel like here for me. Temple to temple, eyes, kind of like this inner corner of my eye. Ugh. All right, does anyone need more time on this slide? It's okay if you do. So migraines, yes, migraines are totally different than headaches. People who get migraines know this. People who don't actually get migraines often think really bad headaches are migraines. I wish migraines were like really bad headaches. I get a little bit of everything when I get migraines. My migraine almost always starts at the back base of as it progresses. My migraines typically last me somewhere between 8 to 12 hours. I've had some that lasted over 24 hours, but usually it's about half a day worth. I'm some weird combination of extremely hot. Like, if I take my temperature, I actually have a fever, but I'm also like shivering and cold. I'm very sweaty. Vision does get blurry. I'm extremely sensitive to all sounds and light, any type of movement. I'm extremely nauseous, and I always end up vomiting when I have migraines. I think that's about. Sometimes I get a little bit of vertigo, like the room is spinning. It's wonderful, great. Angel said, is that like when animals can sense the weather changing / natural disasters? Well, I think animals, because they are outside all the time, are just way more in tune. I don't know if I would say that they can sense pressure changes, right? But they are more in tune with feeling and noticing changes of temperature outside, with changes in wind, the amount of wind, the force of it, the patterns of it, precipitation. And those typically all lead to knowing that certain storms are coming. Yeah, I'm sorry, Fado. I will say, luckily, through eliminating everything delicious out of my diet, I found that almost all of my migraine triggers were food related. One of the number one things that causes migraines if you get migraines is MSG, or monosodium glutamate. And if you start looking at ingredients and probably a lot of delicious food and snacks that you eat, it's in like everything. So almost any type of snack that's cheese flavored, or has like a salty mixed flavor. So like all Doritos have monosodium glutamate, Cheetos have it, flavored chips have it. I know. No, I understand, Fado. A lot of cheese dips have monosodium glutamate in it, but it's a huge trigger. And a lot of fast food, if they use seasoning salts, a lot of Asian food uses it. So unfortunate. Jayla, you said you had a question. Can you smell when the seasons change? Yeah, I think fall has a different smell. I think spring has a very unique smell. Winter is kind of void of smell to me. Summer is just, smells thick. You know what I mean if I'm saying that. But yeah, if you get headaches regularly, I would recommend trying to like start testing out, like removing one ingredient at a time and seeing what happens. You might find that you know exactly what causes your migraines. Even though you're right, Fado, like that food is all so delicious. Like vinegar and MSG are my two largest triggers. And vinegar is also in like almost all sauces. I used to get like 2 migraines every single week. Every other day I would at least have a headache. And even though all that food is delicious, I would rather not eat the food than go back to having migraines every single week. That was terrible. All right, so let's look at our recombination frequency example here. It says you are told that there are 3 genes we're looking at: gene A, gene B, and gene D. You're told that the recombination frequency, so how often the chunks are swapped between B and D, is 5%. The amount of times the genes are swapped between B and A is 30%. And the amount of times the genes are swapped between D and A is 25%. So if we visualize then that this big purple chunk here is a chromosome, right? Because our genes are in our DNA, and during crossing over of meiosis, what's swapped are the chunks of chromosomes, which are just wound up DNA with the genes. So here I got my big chromosome. If I have a smaller frequency, that tells me that the genes are closer together. If I have a larger frequency, it tells me the genes are further apart, okay? Now I purposely chose A, B, and D because by nature of how we are, we want to sequence them in alphabetical order, but alphabetical order has nothing to do with this, okay? So because there's only a 5% frequency of chunks being swapped between gene B and D, that tells me that they're likely closer together. Gene A and B are the furthest apart, and they are here when I visually drew it as well. Okay. And then gene A and D are 25% apart, so they're still pretty far apart. Okay. And so we have that represented here. Again, the reason this is useful scientifically is because if I can then sequence different genes, I'm then able to target which chromosomes in a karyotype and which area in a chromosome carries which traits or genes. And that really is useful when we're studying and trying to understand the causes of different health defects, health disorders, health diseases, if they're genetic, where they're stemming from, and then be able to try and brainstorm and create solutions to these problems. All right, so I will pause here for a second. Whoa, did you know one of our hall monitors is 94 years old? Listen, I knew Leona was she's old. Okay, I knew that. But 94. I don't think I processed like, dang. Well, okay. Doing good for 94, shoot. Jayla, I definitely would not be able to work in a high school at 94. I just feel like once you get into your 80s and beyond, like, do you care anymore? What I mean by that is like, I would just say whatever I'm thinking. I would just do whatever I think is right. I don't know if at that age I would have enough self-control or care to like follow rules and policies and be nice to children. All right, does anyone need more time here? Yeah, there you go, Eli. 94 Roseman's gonna be at home. But I do always joke that I'm gonna have to teach till I'm like 97. You know, because what is a savings account? Ooh. Well, Fado, but the thing is, you're in high school, so you shouldn't know her. I'm twice your age and I don't know her, so. Hey, Brian, just let us know when you are done. I'm so hungry. Yes, Miles. Bananas. When the more you start to think about it, the crazier it is to think about all that she's lived through. Okay, here. So first, I need this text box to be forward. Yeah, no, my grandma Moy, my mom's mom, is turning 94 this summer. So yes, accurate. Okay, so here is a College Board question. Okay, it says, 4 trials measuring recombination frequency between gene R and gene L were conducted, and the results are shown in Table 1. The mean map distance between gene R and gene L is closest to which of the following? So things to first remember here. A percentage originally comes from a decimal or fraction value. Okay, we represent it as a whole number because we conceptually understand what that means, but a percentage is just a fractional amount of something. If it says mean, what does mean mean mathematically? Okay, calculate the average. How do we calculate the average of anything? How do we calculate the average of anything? Okay, add all values together. And then divide by how many values you have. Okay. So you guys want to calculate the average here of the recombination frequencies. Great. So we will go 0.24 plus 0.23 plus 0.26 plus 0.29 divided by 4, because we have 4 numbers, equals 0.28. So the answer would be A, 0.28 map units. This is one of these types of questions where this is such a small detail of the unit. We have some of these in every unit where it's like we only covered it once. I would not stress over that type of stuff for the AP Bio exam. The reality is you're going to get to the exam, and for most of you, you are not going to remember everything. You're not. And that's okay. Okay. So we want to start focusing, though, on these types of things where we spent minimal amount of time and notes on, likely means minimal questions, if any. So those are the types of concepts we don't want to stress about leading up to the exam. All right. Now, our second topic for today are pedigrees. So a pedigree is a family tree that we can use to analyze and determine family members' genotypes and the probability of passing on traits. In a pedigree, we have a variety of shapes, some that are shaded in and not, and these shapes and shadings mean different things. A circle in a pedigree represents a biological female. A square represents a biological male. If the shape is shaded in, it means they have the trait or the phenotype or the physical expression of whatever we're talking about. So this could be a disease or disorder, which is usually, I have a fuzzy in my mask, and I'm trying to, since last hour, it's been bothering me. If it is partially shaded in, it means the person is heterozygous for the trait or they're a carrier. If the shape is not filled in, it usually means that they do not have the trait. Now, sometimes they like to just mess with your brain and they completely switch all that up. If they switch it up, though, they will give you a key that lets you know what the colors mean. When I read a pedigree, you read it top down, kind of like a concept map. So the oldest generation is at the top. Think of like grandparents or great-grandparents. And then it would get younger as we go down. So it would be like grandparents, the next level is like parents, and then children, okay, the youngest part of the family. A horizontal line between shapes represents that a biological male and female mated. Now, I say mated, though we would not normally use that term for humans, because we can use pedigrees for any type of organism, right? Not just humans. A vertical line that comes down from a male and female represents their offspring. Ugh. I'm so thirsty too. I have to have some water. I have to. Oh, that was delicious. Okay. I need to ask a favor of you right now. Could you all individually private chat message me to let me know if you are or are not taking the AP Bio exam? So, Nicole, normally, no. And normally no because of Thurston policy. So Thurston kind of has the policy, like if you're in the class, you're taking the exam. And we had that policy, kind of, if you think about it, makes sense, right? Like if you're not taking the exam, why are you taking the class? Because the class is entirely a curriculum prepped toward an exam. Fado said GPA boost, except a lot of big universities remove the GPA boost. So it actually doesn't really help you that much. Correct, Valentina. They do normally. Which is why, you know, if you're going to take an AP class, you still want to, GPA wise, make sure, I mean, bare minimum that you're going to get a C, but that you're aiming for an A or B still because of that. Well, we are for sure doing the online version. Yeah, once again, say no to transmitting diseases. All right. Does anyone need more time on this slide? Again, it's always okay if you do. I just ask before I plow on. Oh my God, that what? I never thought these words would come out of my mouth. Eli, record it. Actually, we are recording it. That water was so delicious, but I need more water. So interestingly, totally off topic, it is growth, Eli. It is carbonated water, but it has nothing else added to it. So I got like a soda stream. Soda water, nasty. And if you've ever worked in a restaurant or food business and you had soda water, nasty. Okay, I don't know what the difference is, but like totally plain water, carbonated, delicious. And on that note, pedigree example number 1. Good question. Purified. Because since I've gotten used to drinking purified water, I've noticed when I go to spring water or even tap water, I can taste the minerals so strongly. And it grosses me out. All right, so here is a pedigree. It says shaded equals Huntington's disease. So they have this disease. Individuals have it or they do not. There are no carriers in the sense of what we know as the term carriers. Okay, but obviously people could be heterozygous. So according to the pedigree, how many individuals here have Huntington's? Okay, so there are 5 shaded in shapes equals 5 people have it. Okay. I'm so obnoxious. I don't need to do all this, but I do. All right, how many males versus females have Huntington's disease? So how many males have it? All right, good. So 2 squares equals 2 males, and females that have it equals 3 circles that are shaded, so 3 females have it. And again, as we're going through this, I know for some people this just clicks right away, and if you're sitting there like, I don't understand how you got that, please ask. So is this trait dominant or recessive, and what would make you think this? Okay, so let's take a second and sketch this out. So we know originally, right? We've got this. Okay, we're trying to figure out what their genotypes are a little bit in order to figure out if it's dominant or recessive. Okay. We know this mom is normal / healthy. Okay. We know this one has Huntington's disease. So now if we go back to this, let's try to map it out and see, you know, so let's say, I saw someone say, let's say it's dominant. Okay, if it was dominant, then that would mean this could be AA or AA, right? And this would be lowercase. I say AA or AA like I distinguish anything there. Okay, so possible offspring here could either be all big A, little a, which would be all shaded, or 50-50 heterozygous versus recessive. Okay. How many children do these parents have? Ooh, 5. Very good. So this one here, this one, 3, actually 4, 5, and 6. Okay. Now, it's not 8 because this circle connected to the square, that's who square mated with. Okay. This square, that's who mated with circle. Okay, so they have 6 children. 3 of them have Huntington's disease. Is it different if we flip it? So if I were to flip this and say, okay, let's say, oh my gosh, can we not? This is big A, or big A, little a, and this is little a, little a. Okay, so then either all are big A, little a, which is not shaded, or 50-50 are unshaded versus shaded. Did it make a difference if it's dominant or recessive? Statistically, did it make a difference if it's dominant or recessive here? No, it's not. So we can't tell by those children. Okay. Let's look at these two parents here. So these two parents do not have it. Both of their kids do not have it. We've got the same situation here. Here, most of them do not have it. Okay, most do not have it here. So I would say here, given this, a correct answer would be, you cannot know with certainty if the trait is dominant or recessive based off the provided pedigree. Okay. I would then go on to explain why. Okay. This is because if the trait is dominant or recessive, there is still a 50-50 chance of the offspring either having the trait or not. Okay. However, one could assume that because the majority of offspring produced in the family do not have the trait, that the trait is recessive. Okay. So to me, that's a very complete response and answer to this. Okay. There are some instances where you can't actually give a definitive answer to something. And that's not bad or wrong. It's the reality of a situation. Sometimes in certain problems, you are not given enough detail or information to know with 100% certainty what is happening. So it's good to explain and disclose that. But it's also okay then to say, using what we understand about genetics, right, that because a trait does not show up that often, that it's likely recessive. I would take it probably then one step further and say that recessive traits only show up when inherited without a dominant variation, leading them to be expressed phenotypically less often. Okay. All right. So let's assume it's recessive. What would you say their genotypes are? Okay. So correct, Fadl. So if we assume it's recessive, okay, one person has to be heterozygous, another person has the recessive homozygous variation. Okay, let's think about why, right? If I kind of go over here. Okay, so I know one, if it's recessive and someone has it, one person, right, has to be like this. My other options then are this or this. Well, if I pair this together, all children will look dominant, which means none of the children or offspring from that couple could ever have Huntington's. Okay, so that can't be possible, right? We have some kids that have it. However, if I do this combination, right, we'll have 50-50. 50% chance the kids will have it, 50% chance they won't. Okay. Now, again, if we're saying that Huntington's is recessive, then that means this would be the genotype for the person that has Huntington's disease. This would be the genotype for the person that is normal or healthy. Does that make sense to everyone? Does anyone want to ask a question? Okay. The male grandparent would be homozygous recessive, and the female grandparent would be heterozygous. And then I would explain why the female grandparent has to be heterozygous. Sorry, I wasn't saying that out loud because I already said that out loud. Okay. Again, does this make sense? Does anyone need or want to ask a question? So the no carriers part is trying to tell you that it's not sex linked. All right, are we okay here? Okay, so example number 2. So example, I'm always so weird with formatting. Says researchers hypothesized that red eye color in Japanese koi, a type of fish, is due to a mutation. To study the inheritance of red eye color in koi, the researchers conducted breeding experiments over several generations. The results are summarized in figure 1. Based on the data in figure 1, which of the following is the best prediction of the mode of inheritance of red eyes in Japanese koi? So first, is this sex linked? Okay, so let's try an example here. Okay, so let's look at this couple right here. So let me redraw it. Okay, so we know that this couple has red eyes, and this has, what does it say, black or ebony or something? Okay. So if it's sex linked, this is our female. She has X's. This is our male, XY. Let's start by saying it's dominant, okay? So the female could be X big A, X little a, we don't really know. And the male, okay, would be big A, right, if we're assuming it's dominant. Okay, so this is if it's dominant, then the female would have to have what color eyes? You get one X chromosome as a female from your dad. So if it's sex linked and X linked and it's dominant, then the girl would have to have red eyes. Okay, well, she doesn't have red eyes. So it can't be X linked and dominant. Can't be. Okay. So now let's say, this is going to get so messy. Okay, now let's say it's recessive. So if it's recessive, mom would only have little a's, dad would have a little a and Y. If it's recessive, what color eyes would the offspring have? Red again, right? Okay, so it also cannot be X-linked recessive. And there's no other in between, so it can't be sex-linked, okay? Now, a lot of kids will automatically assume it must be sex-linked or X-linked, or else why did they tell me male and female like this broken down? They'll see the broken down of male and female and be like, it must be sex-linked, okay? But it can't be. Not, hello, hello. So that means it cannot be C or D. Okay. So autosomal is not a sex chromosome. So an autosomal chromosome is just a chromosome that's not a sex chromosome. Okay. You know, scientists, we like to give things unnecessary words. So Fadl then said autosomal dominant. All right, explain yourself, Fadl. Why do you think autosomal dominant? Okay, let's go back to this couple right here. So if it's autosomal dominant, okay, that means both parents here have to have at least one big A, right? If it is autosomal dominant, you're not necessarily, no, don't doubt yourself. If you're autosomal dominant, okay, could I have kids that have totally different physical features than me? Look at the genotypes I wrote. Don't doubt yourself. Just take a second and think about it. Yes, good, Valentina. So if both parents look dominant with red eyes, but are actually heterozygous, it could just be that both times that they had kids, they all inherited only the recessive variation. Okay, so a lot of times for these pedigree problems, I think it's more useful to look further down in the pedigree at an isolated family as opposed to the broad spectrum, so you can make more specific predictions about genotypes, okay? So this is autosomal dominant. If both parents in the second generation have red eyes and are big A, little a heterozygous, could have little a kids. So the answer equals A, autosomal dominant. All right, does anyone have a question about that problem? Okay. Let's look at our last example here. Our last example, oh my God. I was like, Kaylin, you're not going to need to be able to see this. It's okay. What is vision? So now I have to get like inside my computer screen. So just wait till I get this close. Wait, do you actually like for real have this like diagnosed, or like this just happens to you? Gotcha. All right. So it said, I hate you, Valentina. All right, several members of a family are found to involuntarily sneeze when exposed to bright light, such as sunlight. Following analysis of the condition in the family, a doctor predicts that the symptoms have an underlying genetic basis. A pedigree of the family is shown in figure 1. So if they are shaded in black, they sneeze when they are exposed to bright light. And if they are not, they do not sneeze. So for this condition, which of the following modes of inheritance is consistent with the observations? Is it autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive? Well, let's sketch each one out. Okay, so autosomal recessive. Let's start here. Autosomal recessive means we see in the pedigree, right? Both parents sneeze in bright light. So if the trait is autosomal recessive, that means both parents would be like this, which means all of their kids would be like this, right? Okay, and we know that that's not what happened. They don't all only have kids that sneeze in bright light. So cannot be choice B. If parents were homozygous recessive, they could only have little a kids, or kids that sneeze with light. Give me 1 second. Can you guys hear me? Okay. Miss Badham wants to see you in the ESC. Yep, both of you. So you guys can start packing up and walk down there, and then after she talks to you from there, you'll be dismissed. Okay, thank you. Okay, autosomal dominant. So both parents have it, which means it could be like this, or could be like this. Well, again, if both parents were homozygous dominant, then the only kids they could have would be kids that sneeze in the light, okay? If we had it like this, they could have kids that were like this, or kids that are like this, or kids that are, oops, like this, right? Okay, so yeah, can't be that. Okay, let's keep this on the table here. All right, another one was X-linked recessive. Okay, if it's X-linked recessive, I've got mom and I got dad, and all the X's have a little a. Well, again, if it's X-linked recessive, that means all the children would be having the trait, right? But we know all the kids don't have a trait. Ah, very good, Angel. Yes, 100%. One parent could be heterozygous and one could be homozygous dominant. But if we do one parent, okay, so let's look at that example. Side note, so we know it cannot be X-linked recessive. Let me type this in right now before I answer Angel's thing. Oh my God, Google, I didn't ask for all that. Very good, Valentina. So that's exactly what we're going to draw out. So if we draw out Angel's scenario, if I have this crossed with this, these are my options of children. Would any of the kids sneeze in light? No. Okay, so it can't be that. So we're still thinking, is this a possibility over here, two heterozygous parents? The last one to test out is sex-linked, and it's dominant. So dad has to have it, mom has to have at least one dominant version, okay? Okay, so here's if it's X-linked and dominant. Very good, Fadl. So Fadl said, if it's X-linked dominant, then all of the females have to have it. One could be a carrier, right? She might not be homozygous dominant, but they still all show, which means they all sneeze in light. If I go back to my pedigree here, one female sneezes in light, but one female child does not. So that right there gets rid of this possibility, which means the only one that is possible is for it to be non-sex-linked and dominant. Okay, so it cannot be choice C, because then all female offspring would have the trait, but not all female offspring actually have the trait, according to the pedigree. So the answer is equal to choice A, right? They are likely, both, not likely actually, both parents are heterozygous, big A, little a, allowing them to have offspring that have the dominant trait and some that do not. Okay. So first, remember I said we could go over, which is perfectly fine. So we can go over to finish the Kahoot. If you need to leave at 12:25, that's fine. If you need to check out with your advisory teacher, just email them. Let them know like we're reviewing, and I can't imagine a teacher not being okay with that. Okay. If you need to leave period because you have something else scheduled, like at the end of school to go to, the Kahoot is very strongly based off the Quizlet flashcards. They are linked in Canvas at the start of the unit, right here. So if you're not able to be here for the Kahoot, you can always get the same practice by using the Quizlet flashcards, okay? All right. Oh, I gotta make this smaller. Oh my God, my kids are gone. I can eat. Shh, you already know I'm about to eat. Oh, what is? William, you are good. Have a good afternoon. Melon. I like it. Oh, that's okay. Yeah. All right, anyone else I'm waiting on? All right, let's start. What is an allele? Bam. Variation of a gene. Good. What does the law of segregation observe? All right, good. That sister chromatids separate so that each sperm or egg cell gets one allele per gene. And it's sister chromatids because it happens during anaphase of meiosis 2. What does the law of independent assortment observe? My microwave, sorry. Why is this so hot? Okay, I got my food. So sister chromatids separate randomly. So that means they're dominant, recessive, male or female traits, doesn't matter. They just go whatever way. Guarantees a mix up and increase of genetic diversity. Now remember, as we go through this is not saying that like if you get all these right, you're gonna get 100% on the test Tuesday. As we now know, for the large bounty of content you have to know and learn, that is just one small stepping stone to doing well on these exams, right? But bare minimum, you have to know the content and the vocab words to be able to make sense of the questions they are asking you, right? So it's kind of this combination of like ACT SAT level type reading, comprehension, and writing skills, combined with scientific method skills, combined with bio stuff. All right, which of the following is true about dominant traits? Okay, they do show up more often, -5. I'm sorry, who chose that? You probably hate me right now. You're like, ugh, it's Rosama. How could you ruin my streak? Okay, which of the following is true about recessive traits? Good, it's only expressed if there's no dominant present. So the person has to be homozygous dominant. And if it's X-linked, they only have to have the recessive version, right? If I'm a male. What does homozygous mean? Good, 2 of the same alleles. It's not necessarily always dominant or recessive. It depends which variation is inherited. What about heterozygous? Good. So heterozygotes always look like the dominant variation, right? Because if you have a dominant, it's going to show or be expressed. I'm going to also turn my camera off because I'm about to eat and it's going to get messy. All right. Your names. Identify the genotype for homozygous dominant. Good. 2 capital letters, 2 dominant variations. What about, did I do this again? Oh, no, it's the right one. What will homozygous dominant look like? I hate myself. Yes, look dominant. You guys are so fast, I don't even have time to eat. What is the genotype for heterozygous? One of you politely waited longer. Thank you, though. What is the phenotype for heterozygous? Good. It's the genotype for homozygous recessive. Good. And what would homozygous recessive look like? Smelling, moving up. Incomplete dominance. All right, good. So the stuff on the right would be for normal Mendelian genetics. The stuff in the left or red would be for like incomplete and co-dominance. What's true about incomplete dominance? You gotta give me a minute. I'm eating. Very good. So remember, in incomplete dominance, the heterozygote, all the traits are dominant. And it's called incomplete because the dominant traits are both only partially expressed at the same time. So because they're only partially expressed, it looks like a blending of the two traits or whatever the in-between might be of the two traits. What about genotypes for codominance? All right, good. Again, remember the ones with lowercase letters are going to be for Mendelian genetics. Codominance and incomplete dominance only have dominant variations. All right, good. So remember in codominance, co means together. The two dominant traits are fully expressed together at the same time. Jason, you are good. Have a good day. Which causes not a blending, but both to fully show up simultaneously. So it often looks like spotting, stripes, things like that. Yeah, Fadl, there's 40 questions. Devin, you are good. Have it.
You too.
Is not true about sex-linked traits. Who's here? Dante, you are good. Have a good day. Good. So sex-linked traits are usually on the X chromosome. They are usually recessive, and people can be carriers. Female sex chromosomes. This should be easy. Good. XX. All right. Male sex chromosomes. What is recombination frequency? Asia, you're good. Have a good day. My mouth could not have been more full. What does recombination frequency tell us? All right, good. What does a circle represent in a pedigree? Mia, you're good. Have a good day. Good. Female. But you can never guess what the question is. What does a square represent? Male. Ooh, Schmelin taking the lead over Fladoodles. What does a horizontal line represent in a pedigree? Fadl, I actually switched some of them like that because I wondered if some of you were going to be thinking that. Anaya, you are good. Have a good afternoon.
You too.
What does a vertical line represent? What is the equation for chi squared? Good. You know, the only thing wrong in the green one, right, is that it's over the observed. How do you calculate degrees of freedom? All right, good. So number of rows minus 1. Blue and yellow. Blue says divided by. If you have a p-value below 0.05, okay. I'm gonna try not to talk with a full mouth of chili and chips. Your results are significant. So it's 0.05. It says we have a below 5% chance that there is not a connection between data, which means it is likely that the results you got are directly related to whatever independent variable you manipulated. So you want to have a p-value below 0.05. What if the p-value is above 0.05? Your results are not significant. So this doesn't mean with certainty that your results are wrong. It just means given those results from that experiment, currently there is no correlation between your results and what you manipulated as the independent variable. So usually when you get results like this, if you strongly believe that there is a correlation, then it's time to either go back and retest it or figure out different ways to test it. What is a control? All right, good to whoever chose yellow. Very good, Fadl. Wait, you said red would be constant. No, green would be constant. So yellow, it's an entirely neutral setup where we usually don't add or change whatever the independent variable is, purposely causing no results to happen. The whole reason we have a control is to purposely see nothing happens. And you might be thinking like, that's the dumbest thing I ever heard, but it's not. Because then if we do our experiment with an independent variable and nothing happens like in our control, that tells us that the independent variable with certainty had zero connection or correlation to any part of the system we are testing. What is a constant? So, right, these would be things that are like the same. You use the same species of bacteria, you use the same type of antibiotic, same concentration of antibiotic. You provided the same type of agar medium for the bacteria to grow in. Okay, things that we keep consistent between the setups to minimize the number of variables that could be affecting our results. Which variable goes on the x-axis of a graph? Good. What about the y-axis? That should have been a duh after the last one. What is an independent variable? Good. What's a dependent variable? Good. It's what happens as a result of what we played around with or adjusted. What is a null hypothesis? Good. Do you want your null hypothesis to be true? Okay. When a question asks for evidence, you should keep that in mind. If something says justify, what does that mean? Good. All right, let's see here. Pancake coming in strong. Angel, wonder who that is. Who we got here? Ooh, I didn't know if it was gonna be Fladoodles or Schmelin. Okay, but Schmelin and Nikki are runners up. Good job, good job. All right, we are done. I'll probably send you an email later today, or yeah, I assume Fladoodles was Faddle. I'll probably send an email later today or early tomorrow with just some tips of how I would approach studying for the exam. Your exam again is next Tuesday. Anyone have any questions, preguntas, etc.? Okie dokie. Well, you know the deal. Email me if you're having a life crisis about something. Yep, Faddle, you'll do the FRQ during class and multiple choice afterwards as homework. All right. Nope, you do not. Okay.